We've had a lot of interest in this topic, which is pretty exciting. And in fact, more people have signed up for this one than any other yet. So we're going to make the most of it. The way this is going to work is Alec is going to give a presentation where he has a slideshow, because some of the material is pretty, I don't know, complex. But he is going to pause during the presentation to address some questions. But instead of just raising your hand or asking the question, we'll ask everybody to be on mute and type your question into the chat. We'll monitor those questions, and Kelsey will, when we come to a natural break in the presentation, Kelsey will read off some of those questions and allow Alec a chance to answer them. And then at the end, we'll have a chance for more of a Q&A, so to cover any questions that he didn't answer during the presentation. So this should be a discussion, like always. Let's have a conversation, but Alec is going to get us started. And I keep calling him Alec, but his name is Alexander Ernst, and he is from Cornell University. He graduated with a Bachelor of Science in 2017 and has since been working in the biomaterials and cell therapy laboratory under Minglin Ma, his professor. So I will let him continue to introduce himself, and then we'll get started. And I think we're all pretty excited. So Alec, please take it away.
Appreciate it. Thanks for the introduction, Rob. You guys can all call me Alec. No one calls me Alexander except my mom when she's mad. So please stick with Alec. So thanks again, Rob and Kelsey and Gales Creek, for inviting me to do this. I'm Alec. I'm a PhD candidate in biological engineering at Cornell. I'm actually defending my thesis 13 days from now. So if you guys want more technical version of this talk, you can feel free to make the trek out to Cornell in 13 days. What I'll be discussing today is what our lab research is, which is cell encapsulation as a therapy for type 1 diabetes. But first, here's an introduction to the Ma Lab. On the right over here is our leader, Professor Minglin Ma. I'm over here on the left looking a little shaggy, and I'd be remiss if I didn't also introduce a close friend of mine and my research partner for the last 5 years, a postdoctoral candidate, Dr. Long-Hai Wang. I'd say many of the most brilliant ideas in our lab come out of this guy's noggin. Cornell is in Ithaca, New York, a town in central New York, which is beautiful 50% of the year. And today was one of those days, but normally we're not as fortunate to get the weather that it looks like in the picture. So here's the agenda of what I'm going to talk about today. First, describe what is cell therapy, and then what is cell encapsulation, what are the major challenges with cell encapsulation, and what are our strategies to overcome them. And as Kelsey mentioned earlier, so I've talked a bit with Rob and Kelsey and Kendra, and I know just from those brief conversations that there's a lot I can learn from you guys. And so as much as possible, I'd love to make this a discussion. If we don't mind for a second, I'm going to check to see if there's anybody else in the waiting room. Let me figure out how to do this. Oh, here we go. I think it was just one. Okay, I think that sound will notify me if any more people are in the waiting room. Sorry for the little pause. Okay, what is type 1 diabetes? Again, I'm sure all of you are much more familiar than I am, in a more intimate way at least, of what type 1 diabetes is. But I still think it's important to start out with this slide just to set the stage for the rest of the talk. So what type 1 diabetes is, from our research perspective, is it's the immune destruction of the pancreatic beta cells. Beta cells, of course, are insulin secreting cells, and they're located in little microorgan cell clusters called the islets of Langerhans. And they're called this because their discoverer, Paul Langerhans, thought that they resembled islands, in sections of pancreases. And these cell clusters, they're about 500 microns thick, so 0.5 millimeter. A good reference is the size of your thumbnail, maybe a little bigger. And beta cells are one of many cell types in an islet, definitely the most common. And of course, what they do is they sense the amount of blood sugar is in your system, and in response to that, secretes insulin, which makes its way to the rest of your tissues, telling those tissues to make food, to absorb food as fuel. And of course, type 1 diabetes is the absence of this. So the fundamental concept behind cell therapy is to reconstitute nature's way of controlling blood sugar. What this looks like in practice is an organ donor donates to the pancreas. The islets are isolated from this pancreas and then are purified and put into a solution and then infused into the liver. It turns out that the liver is actually a really good place to put the islets because just like the islets containing the beta cells, which sense and respond to glucose and secrete insulin, the islets there about the size of your thumbnail. So are channels inside the liver. So they kind of self-sort. They go throughout this channel network in the liver and they're self-sort. And this channel network is filled with blood. So just like an islets in a non-diabetic individual, the islets will be exposed to blood. We'll see how much glucose is in there and can secrete blood in return, or insulin in return, excuse me. So this concept was known for many years before the year 2000, where it first became actually a successful therapy. And this conversion happened due to the insights of this handsome gentleman over here, Dr. James Shapiro, who the Ma Labs actually had a great pleasure and privilege to collaborate with. What he believed, and he had evidence to believe this, was that many of the cells, once infused into the liver, would die quite quickly. And so he believed that if you just increase the amount of islets you give the patient, taking islets from 2 or even more donor pancreases, you could achieve success. And he also assembled an immunosuppression cocktail, which prevented those cells now residing in the channels of the liver from being destroyed by the same immune mechanisms that destroy beta cells in a type 1 diabetic patient. So today in Canada and the European Union, this procedure can actually be done by doctors and surgeons, and it's covered by state healthcare. To date, I believe over 1,500 patients have received this procedure. In the US, unfortunately, there's a regulatory artifact which classifies islets as a drug instead of as an organ, and thus it needs to be done by the private sector. It was only until last year where a company called Cell Trans, if you want to look into this further, received permission to do this procedure. And in consequence, islet therapy is very limited in the United States. So what has become clear in the last 22 years of islet therapy or cell therapy is that it's much better than insulin therapy or hormone therapy in general, even the most advanced kinds. So what these traces are blood glucose levels over about a day in a patient wearing an artificial pancreas system. You guys may be familiar with these as the closed loop system. So continuous glucose sensor is algorithmically coordinated to an insulin pump where it infuses insulin through a catheter or another means. And this is before and after treatment with the closed loop system. And you see that there is quite a marked improvement in glycemic control, but it doesn't come close to the glycemic control that can be achieved with islet transplantation. Here are the blood sugar traces throughout a day in a recipient of islet transplants. It's much better. You can just see by the width of the bands. So to summarize, there's been a number of amazing achievements with cell therapy. The first is the near complete abrogation of hypoglycemia. So islet transplant recipients almost have no incidence of hypoglycemic episodes, which I'm sure all of you know better than I do is really remarkable. And also at 3 years after the transplant, 44% of the patients don't need to administer any exogenous insulin at all. And at 10 years post-transplant, even though there's some graft decline, some of the function gets a little worse, there's still a reduction in the amount of exogenous insulin that's needed in 80% of the patients. These are remarkable achievements. But there's still a number of challenges with this procedure, hence why only 1,500 people have received it in the world today. One of those reasons is the dependence on donor pancreases. And again, more than one is needed per recipient. There is new hope, though. that the dependence on donor organs may be eliminated. Many of you may have seen this article that came out in the New York Times, which describes a patient who received an infusion of stem cell derived islets. So instead of taking the islets from a donor pancreas, they're essentially grown from new in the lab. And his diabetes was completely corrected. And even though it's just one patient and only a short time after the procedure, this result is really encouraging and it's really remarkable. But there are other challenges with islet therapy alone, and that's the gradual decline in graft function. So over time, some of the immune responses that destroy the beta cells in a type 1 diabetic patient in the first place kind of overcome the immunosuppressive cocktail. And the second or third challenge, I suppose, is the requirement for patients to receive lifelong immunosuppression. And this immunosuppression, it's I don't know how to qualitatively describe it, but it's the important point is that it limits the scope of patients which doctors feel comfortable prescribing the therapy to. So just to recap, cell therapy, it's a concept that instead of delivering insulin or hormones into the body, you can deliver insulin-producing cells into the body permanently. Also, islet transplantation, a cell therapy, is better than insulin therapy at controlling blood sugar levels. And third, why this isn't widely available to everyone and isn't a successful treatment yet for huge amounts of patients is because it still requires patients to take chronic immunosuppression. So I think now would be a great time for questions.
Here is one, Alec, actually two. So the first one, just for those who can't see the chat, asked, are there articles about the 1,500 people who have received the therapy?
I can share some academic articles. How much sense they'll make to a non-academic audience is, I'm not sure, because, and even, in this talk, if I say something that you guys don't understand, there's a good chance, I'm just head down buried in the academic literature in the lab. And so sometimes I don't even recognize when something is as understood by everyone or just researchers in the field. And so I can definitely share some, and some really excellent, the doctor, the handsome gentleman that I showed a picture of, he's actually a really great writer, even in an academic sense. And so I can share some articles by him who's described the progress of this treatment over a while.
Awesome. The second question is, how about using stored umbilical cord blood? Would you still need the immunosuppression?
Yeah, I'm actually not familiar with this therapy. What I'll say is that if you put something from someone else into the body, you generally need to find, your body is just remarkable. It's just so creative at figuring out how to get around, at detecting things that are not supposed to be there, and even detecting things that should be there, but is poorly trained to destroy. So I would imagine so, but I don't want to say anything further because I'm actually not familiar with this particular vein of research. Leanne's question.
Perfect.
Chronic immunosuppression seems like a big deal. I guess my question is, might that change at some point? So this is just a perfect segue, because this is really what our lab is trying to do, and our field more generally. As I mentioned, so this is where cell encapsulation comes in. Cell encapsulation, for a non-academic audience, it probably doesn't mean a lot. But the concept of cell encapsulation is that you can take islets, which contain the insulin secreting beta cells, and you can wrap them in a semi-permeable material. What the semi-permeable material means is that it keeps the immune system out. So the two main agents that destroy beta cells in a type 1 diabetic individual are cells and antibodies. Maybe after the experience with the COVID-19 pandemic, we're all familiar with antibodies now, but basically a molecule and cells will attack the cells. But you can keep them out with this material. But it's semi-permeable, so it does allow oxygen and nutrients, and of course, importantly, glucose to go into the device, into the material, permeate through it. Likewise, insulin and other hormones made by the islets to leave. So this is the concept for obviating the need for immunosuppression. It's one strategy, which our lab focuses on, which I believe is promising. So what the semi-permeable material is a really novel class of materials called hydrogels. And they're kind of like the boba balls of a bubble tea. They're kind of squishy gels. And like your organs, they contain a lot of water. Why? And this is why hydro is in the name. In particular, the 1 Hour Lab really uses a lot and has really found to be quite a remarkable material is a seaweed-derived material called alginate. And it's, most of you are from the West Coast. I grew up on the West Coast and I've swam in the Pacific Ocean and on the Oregon coast. I know that brown seaweed occasionally is, by the shore. And that's really kind of what the material is like. It's kind of like, it's like seaweed without the brown stuff, even in texture and feeling and consistency. And the islets, they really like it because, again, it kind of resembles where they would normally live in non-diabetic pancreas. It's just a big water sac. And it's kind of fine with the body too, because again, it's all water. Now, here's the chemical structure. You guys don't need to pay attention to the details, but if you guys just close your eyes and remember 10th grade chemistry, you may recognize, you probably don't, and I don't blame you, that these look like sugar. And it's because they are, but unlike your sugar, which critically, it'll go into your tea and your coffee, it'll dissolve. These will dissolve too, but the bonds between each sugar molecule are really strong. And so instead of just going into the liquid, they form this gel, which is like a solid material that's still squishy and just a happy home for the islets, which critically does these 3 functions. It keeps the immune system out, lets oxygen, nutrients, glucose in, lets insulin out. There's 2 main structural approaches for islet encapsulation. The first is microcapsules. And these are just like the boba balls. They're just like the little balls in the tea. They're about 1 millimeter big, sometimes smaller, sometimes a tiny bit larger. They'll contain 1 or a handful of islets. And you can just inject them into the abdominal cavity. The advantage of this way to do it is that it's easy to implant. You just do it by injection. It's really minimally invasive. They're also really easy to make. Like we can just make them for food applications like your bubble tea. But the cons are that they're difficult to remove, if not impossible to completely remove. And the other thing is they tend to clump together. So when you put them and you infuse them into the abdominal cavity, and then they'll just kind of bunch up. And so then they're not really spheres anymore. And now some of the balls at the center of the clump, they're really far away from the nutrient source, and it takes a much longer time for them to sense and release insulin. Why retrievability important is it's really just a safety concern. So Sigilon is a company which makes a microcapsule for hemophilia A. A similar concept to what we do for type 1 diabetes, but they do it for hemophilia. And recently, one of their patients experienced an adverse event. The details of the adverse event aren't that critical for this, but unfortunately, this patient's okay now. But it would have been really nice to overcome this problem. They could just remove the system, but they can't because there's just capsules all over the place. So the other kind of approach, the structural approach for islet encapsulation is macro scale devices. So these are just like a big slab or a big tube that contain about centimeters big, so like maybe the size of your hand or something. And they contain many hundreds of thousands of islets, just all in one system. So the advantage there is that it's safe, you can retrieve everything, and you can also augment with other features, which is just we'll talk about later in this. And but the downside is that now the cells are close to each other, the islets are close to each other, and so like you keep your kids in the car too long, they'll start fighting. It's kind of like that. They have to compete over whatever resources are available to them. And the other thing is it'll require more complicated surgery. So whereas the microcapsules is just an injection, this you need to actually, it's maybe like just putting a Band-Aid but underneath the skin. Okay, another recap. The concept of islet encapsulation is the way to address Leanne's question, which is you take stem cells, or excuse me, you take cells, which can be stem cell derived. Some people have tried pig islets. Some people, or most people, a lot of people have tried just human cadaveric islets. You put them inside a device which protects the cells from the immune system, and then you put that in the body. And just a quick word on where you can put it in the body. There's basically two options. People have tried more, but there's basically two. That's underneath the skin, and then in the intraperitoneal space, which is just the intra-abdominal space. So I guess it's the way to describe it. It's just like a fluid sac that all your other organs, like your liver, your stomach, your intestines, they just hang out in this sac called the fluid sac, called the intraperitoneal space. The advantage of under the skin is, like, you guys can imagine it now. Just it's just more accessible surgically. It's right there. But there's worse exchange of insulin and nutrients. There's actually not as much blood vessels underneath the skin than there is in the intraperitoneal space. And the reverse is true for the latter. All right, now I think is a great time for more questions. Yeah, what is the life of the encapsulated islets? That's a great question. And to be honest, I'm not sure I know the definitive answer to that, but I would imagine it's similar to the lifetime of if it's from a donor, it's probably similar to the lifetime of that donor. I would say it's on the order of years or decades, but there's other challenges which may reduce that. So there's external challenges which may limit the function, or over time gradually diminish the function of the islets, independent of what their natural lifespan is. I'll say that the natural lifespan of the islets doesn't seem like a problem. There's other challenges which are more urgent, which we need to address more urgently. Maybe this will be a problem, but it hasn't been in our case. That's a great question. So Laura asked, after the encapsulated islets died off, could you just add more? So in islet transplantation, without the encapsulation, that's kind of what they do. They'll just keep on infusing islets until they see a result. And you can do this also. You can just add another device and another device and another device until you have enough. What that adds, though, is, like, ideally you don't want to do that. Ideally, you just want to get one implant, or maybe one implant every 10 years, which treats it. And this is really where you guys who have a more intimate understanding or intimate sense of what you would accept for yourselves or for your children as a solution. But there are a number of people, and us included, we've thought about ways. Okay, so let's say the graft slowly starts to fail. Can we now replace it? And I think it's feasible. It depends on the system, but that is a great way you can address it. Okay, so far everything seems reasonable except for the lifelong immunosuppression cocktail. Would collecting and using a person's own stem cells to recreate the islets, that's an excellent idea. So this has actually been explored. You can take stem cells from your own. So in, I don't know, the early 2000s, researchers figured out how to basically take a skin cell, turn it into a stem cell, and then turn it into whatever cell you want in the body. And in this way, you wouldn't even need encapsulation. So the point of encapsulation, and that's putting it in the seaweed material, is to avoid the need for immunosuppression. In our animal models, we don't need to use immunosuppression. And we can project that in a patient too, we wouldn't need to do that either. People have tried this. It's called auto transplantation. So just transplanting cells into yourself. The limitation with that is it's a little pricier, and you probably wouldn't. And the other problem is that you would have the same mechanism that destroys the pancreatic beta cells in a type 1 diabetic individual is still present. And so I suppose you would need a way to protect them. But that is an excellent, that's anticipating what a lot of research in the field actually does, which is pretty cool. There's a question, what are the impacts of long-term immunosuppression? I have to apologize, I'm not a doctor. I really don't know what the long-term effects of immunosuppression are. I know it's suggested to increase the chances of cancer because your body can't fight certain just abnormal functions. And I know in the short term, there's higher incidence of ulcers in the mouth, but I'm not really sure what the long-term effects of immunosuppression are. I've actually really wanted to answer this question, but I want to talk to individuals who've received the procedure and can describe it. I'm not familiar enough with that literature to tell you exactly what the long-term effects are, unfortunately. Okay, so Julie now clarifies the stem cells from the umbilical cord of the patient. Yeah, so the stem cells in which the gentleman in the New York Times piece received, he received umbilical stem cells, but from another individual. From the patient, we wouldn't even need the umbilical stem cells. You can actually reverse engineer cells from the skin now. Could you avoid immunosuppression if you just immunosuppressants if you just kept on injecting new islets? I suppose that wouldn't be feasible for a number of reasons. First of all, so now, I mean, maybe with the stem cells, like there's a chance, but I mean, I'm not really sure of the time scale of beta cell destruction in an individual who already has fully matured diabetes, but I would imagine that it's still much better to just, if that was the case, you would just stick with insulin therapy, excuse me. Okay, I think it's time to move on. And again, if you just have questions, you can add into the chat. I appreciate all the questions. They're really great. Okay, so what are the major challenges with cell encapsulation? This is not really an exhaustive list, but I suppose that most could be bucketed under these two categories. The first is the host response, and the second is oxygen limitations. And I'll get to each of them in sequence. So the foreign body response is just this phenomenon that happens whenever you put anything foreign into the body. You don't need to look at all the details on this graphic, but basically just a bunch of stuff that's floating around in the body. When you put an implant in, it'll start sticking to that implant. And then a whole bunch of cells will start coming in, and they'll notice that proteins and other gunk have kind of stuck to this implant. And it's really interesting. Like what they'll do is they'll notice it's foreign, and so they'll all kind of aggregate on it, and then they'll even fuse. They'll basically try to wall it off from the rest of the body. How I describe it is, if you just think about the last time you got a splinter, first it's painful and it's red, and then over time, like this capsule develops around it, and the capsule just tries to reject it, just tries to get it out. This is what happens with the implants too, and the body has just an unlimited amount of ways to just recognize that something is there that's not supposed to be there. It's really incredible. And so the strategy that the Ma Lab, this isn't my research particularly, but the strategy that others in my lab have explored is to just select unique materials and even modify those materials to improve their function. What I mean by this is, okay, so let's take alginate. We make alginate microcapsules and we'll implant them in a mouse. And in a couple of weeks, we'll retrieve those microcapsules. And with alginate, which is compared to just any generic materials, is actually really great at avoiding scar tissue formation. You can still see that there's a couple layers of scar tissue cells on the surface of these implants. So what my colleague Ching-sun, what he thought is that alginate's already pretty great, but you can just improve it a little bit. And so he added these functional groups. We'll talk about those in a second. Don't need to actually worry about the details. He made microcapsules out of his modified alginate, implanted them in a mouse. After a couple of weeks, retrieved them. You see that there was just a significant reduction in the scar tissue formation on the capsules. And what he hypothesizes is that these functional groups, so alginate, like I mentioned earlier, it likes to bind a lot of water. It just really likes water. And that's why we call it a hydrogel, because it's just like water and a gel. And these functional groups just even increase the amount of water that the chemical, that the polymer is able to bind. So what happens when cells are cruising around, they're looking for foreign stuff because that's their job. They'll approach the implant, and instead of seeing something foreign, they'll just see water, and so they'll leave it alone. And so it was a pretty ingenious way to overcome this issue. The second major challenge with cell encapsulation is oxygen. So normally in a non-diabetic individual, let's say you're an islet inside the pancreas. Now blood vessels go all through this structure. So much blood vessels go through the islets that every single beta cell is right next to a blood vessel. But when you encapsulate them, now they're disconnected from the blood vessels. So they're in the hydrogel, and now the blood vessels are just surrounding the hydrogel. And the reason why you can't just invite the blood vessel into the encapsulation device is because then all those same immune components which recognize cells and seek to destroy them would come back. Excuse me. Let's say you could just track a single molecule of oxygen coming from the blood and down into the islets of an encapsulated islet. If you just measured the travel of one, it would take about 10 minutes, which is a remarkably long time for a pretty small distance. This is because oxygen moves really slowly in tissue and water and aqueous materials, just water-like materials. The second problem is that there's not a lot of oxygen around, just floating around outside of the blood. Again, you don't need to look at these numbers, but here is the level of oxygen that a native beta cell will see, pretty high. And then here is the amount that an encapsulated one will see, either underneath the skin or in the abdominal space. It's lower. That's the only relationship that you need to understand from this graph. And so if the oxygen levels are low enough, some of the cells will die, and they kind of die by this process that's like an explosion. So dead cells, they release, when cells die by this mechanism called necrosis, they release a bunch of molecules. They kind of just spray out a bunch of stuff into the surrounding environment. And those molecules are recognized by the immune system, and the immune system will have a better chance at recognizing the graft and being mad at it, basically. The last point is that oxygen is the fuel of insulin secretion. So most cells in the body can take fuel from all kinds of sources. They can take proteins or fats, but just because of the unique biochemistry of beta cells, they can only really use oxygen as a fuel for insulin secretion. So oxygen is really critical to making this whole thing work. What this means in terms of device design is that if you don't solve the oxygen problem, this is back to the kids in the car problem. Like, you need to keep the kids far apart from each other so they don't fight over the limited resources. And you also need to make the device really thin. So remember, I said it's just about, the islet is just about the thickness of a thumbnail. If you don't solve the oxygen problem, you need to also make the device like just barely bigger than the islet itself. And so when you look at the footprint of a device containing a curative amount of cells, it needs to be like a MacBook. And that's just not feasible for anyone. But if you do solve the problem, you can pack the cells much tighter together, maybe even in a couple layers, and that makes the graft a much more smaller, manageable size. This is, I don't have a formal recap here. The formal recap would, I suppose, be there's two problems to islet encapsulation. The first is the immune system. The second is oxygen. And what I'll do next is explain how we've addressed the oxygen problem. So the first way, and this is now getting into the core of Long Hai and I's work, is how do we solve the oxygen problem? The first one is we took inspiration from insect biology. So many insects, they don't, unlike other animals, they don't have a circulatory system that sends blood throughout the body and nourishes the tissues with oxygen that way. What they have instead is this blue structure here, which is inside a mealworm beetle that we just purchased from Petco and stuck inside a fancy microscope. And what you see is these channels here they're just filled with air. And at the surface, in these little regions here, they're just kind of connected to the atmosphere. So gas from the atmosphere, where oxygen is way higher than it is in the body, will just go into these channels and diffuse around. And inside in the gas phase, so when oxygen is in air, it moves way faster than it does in water. This is why you can breathe pretty well in the air and you can't breathe pretty well underwater. And so this network is called tracheae. And so we decided that we can reconstitute this approach for oxygenating insects in an islet encapsulation device. We call this approach, named by my mom, the sonic approach, where we were a little liberal with the acronym. It stands for Speedy Oxygen Network for Islet Encapsulation. But what it is you have the sonic scaffold, which is basically a synthetic trachea. And then just like the insect tissue, you have islets encapsulated inside an immunoprotective alginate hydrogel. And just like the insect tracheal system, oxygen just needs to pass a really thin barrier where it diffuses slowly, but then it gets inside the scaffold, which has this channel network, which allows it to travel really rapidly and go distances way further than it could ever go inside just the hydrogel alone. So here's what it looks like in reality, not in cartoon form. It's basically a white sponge. And even though it looks like a solid, if you do some fancy microscope imaging, you'll see that these networks, the sponge is just full with air channels. And these channels, they're not just pores, they're actually channels that are connected with each other throughout the structure. And this part was actually complicated, but to put it in crude terms, you can just slap on your encapsulated islets. So what this, sorry, I got another notification, but I think it's okay. So what this image here is a microscope image of the actual device. This length here is just a couple millimeters, and it's about 1 centimeter in length. And in black here is the sonic scaffold. And in the gray, when the little flecks, they're alginate and they're encapsulating rat islets. So we can test how well this will work in an animal model. And for that, we use a chemically induced mouse model. And that is basically, we take islets from a rat pancreas, we load them into the sonic scaffold, into the sonic device, and then we put them in a mouse in which we've injected a solution which renders the mouse diabetic. And we can track the blood glucose over time in mice that either received the device with the sonic scaffold or just a device without the sonic scaffold. In animals that received the sonic scaffold, the blood sugar was pretty well under control for most animals for 180 days, whereas the control animals, the blood glucose slowly rose in our random measurements, meaning that over time, the cells inside died. Whereas in the sonic system, they were healthy over this time. I think now is also a good time for questions, because this is one full project that I've skipped over a lot of the critical details, but I'm not sure also if they're that interesting. Are there any clinical trials that patients can sign up for? I do know that there's several. I'm not sure what the criteria are. I do think that the criteria may be adult with type 1 diabetes. And I'm very happy to provide resources for the companies that I know who are actively conducting clinical trials. All right, so now I'm going to discuss the second approach, which isn't as elegant from inspiration, but it's probably way more functional. We call this the inverse breathing system. So if you recall, we have the primary encapsulation system, which is where you put the islets inside some material that allows glucose in, insulin out, and keeps the immune system out. One of the waste products of this system, just like any organism, like me or you or anyone, is that you exhale CO2. These cells, just like you, exhale CO2. So what you can do is you can take, and what we did is take a chemical formula that's used in scuba gear and other technologies that converts the CO2 back into oxygen. So there's a lot of details on the slide. That's all you need to know, is that what this device does is it takes carbon dioxide produced by the cells and then reconverts that back into oxygen, recycling it. And we call this the inverse breathing device, iBED, I guess inspired by Steve Jobs. So what it contains, it's basically like a lollipop in both shape and scale. And it contains the inverse breathing chemical formula, which produces oxygen, and then the islets inside the immune protective hydrogel, and then a gas permeable tubing, which allows the carbon dioxide to go to the inverse breathing formula, and then the oxygen to come back down to nourish the cells. And we did a lot of tests in mouse models where we could show that quite robustly, you could reverse diabetes in the mouse model very confidently. And we also went into pigs. So we did a proof of concept test in pigs. We put rat islets inside the device and then transplanted the device into pigs, which is a really steep immunological challenge. And we did this with a subcurative dose. So we didn't put enough cells in to cure diabetes in a pig, but we just wanted to see if we could make these cells in this really challenging immunological setting survive. And so here's an image on the left taken during the transplantation. It's just on a female mini pig abdomen. This is before it's sutured shut. And then on the right are just images of happy surviving cells after retrieval. So this was really encouraging for us because we showed that this platform can be used to make encapsulated cells survive in a really challenging model in a large animal. So we hope that this is going to encourage us to pursue other large animal studies, and hopefully it'll be a pathway for us to eventually get into the clinic. I'd like to end before we go to acknowledgements with just a humbling reminder, really for me and our group, is that oxygen isn't the only problem. So this device is developed by a company in Israel called Beta O2 Technologies, and their device is called the Beta Air device. It's basically an Oreo hooked up to a snorkel. And this is the Oreo and this is the snorkel. What the two chocolate wafers in this analogy are the encapsulated islet slabs. And in between is just a gas chamber. And it's connected to the snorkel to an oxygen port. And in the patients, this whole system is transplanted underneath the skin. And every day, to make sure that the cells have enough oxygen to survive, the patient puts an injection with a pen into, well, an oxygen pen into the oxygen ports. And so we can definitely say that they solve the oxygen problem, contingent on that you have to still do an injection every day. But I've talked to quite a few type 1 diabetics in the last 2 weeks, and I know that reducing everything to one injection would be a remarkable achievement. But unfortunately, in a small 2018 clinical trial, their device was shown not to achieve any metabolic success. And they believe that this was because even though the cells had enough oxygen to survive, they still had a really strong immune response. So a lot of scar tissue formation on the outside. And this prevented other nutrients from getting in and out. And so that leads us to the road ahead, both what we've learned from our own research and from others in the field, is that what we really need to do now is we've had a lot of solutions to different sub-problems, but now what we're trying to do is combine them into one platform. And the other thing is we need to improve practicality. I failed to mention when I talked about the iBED device that the tip of the lollipop, which supplies oxygen, it can only do so for a finite amount of time. Depending on the system, we imagine maybe 3 months or so. So that would require that every 3 months, you need a minor surgery to replace it. I can't really describe how we've gone about coming up with a solution, but in the last couple of months, these are the problems we've sought to tackle. And I can say sort of cryptically that we've made remarkable progress in addressing these problems, which would really be the first step into making a device that could be clinically useful, like as in actually useful in patients, not just for the mice in the lab. And the last thing we need to do, of course, is to just make these in a robust way. So right now, because we're exploring many different ideas, we use materials that are easy to change and adapt, usually by 3D printing. But a lot of these materials aren't approved for implantation in people. And so we need to, once we settle on a final design, we need to make it in a reproducible way with FDA approved materials. Before I take the last round of questions, I'd just like to, again, acknowledge everyone in the lab. I love this photo. It was taken during the peak of the quarantine period during COVID-19. And I always think it just kind of looks like we're a band cover. And I'd also like to thank the people who funded this project, which are the JDRF, the Juvenile Diabetes Research Foundation, and the National Science Foundation, who funds me personally.
Awesome. Thank you so much, Alec. I can go ahead and read off some of the questions that come in the chat since they might come kind of quick. But if you would like to unmute yourself and ask Alec directly, feel free to just go ahead and either raise your hand or unmute yourself and do so that way. I'll kick it off with Renee's question in the chat. She says, excellent research. It sounds like you are on the way to diabetic recovery. Are we able to follow your research for the road ahead?
Yeah, I mean, I don't know which way to, I'm on Twitter, I guess, but I haven't ever posted anything except one sharing of a research article I published. I would recommend the Ma Lab website. And I'm not sure. I think it is important though to just that we keep on like this is the first time I've done an event like this where I'm reaching out to people who know people with diabetes, who have kids with diabetes, or who are diabetic. But this whole event for me has been really fruitful because it's forced me to just think about how I've done my research, how I frame my research in the context of not just solving the micro problems that are present in the lab, but for actual real people. And so I suspect that we will be doing more of this, the Ma Lab, and hopefully me personally too, more of these kind of events. And hopefully we're invited back. Hopefully I didn't blow it for the Ma Lab.
Not at all. It looks like everyone in the chat said it was great and really great for folks like us who live with type 1 every day, whether it's ourselves or our kids or our family members, to kind of understand how this all works. So any other questions? Again, feel free to unmute yourself or raise your hand if it won't let you for some reason. We'll give it a little bit of time because everyone's just soaking it all in.
Alec, I'll ask a question. It's kind of an unfair question because just about everybody here is a parent of a kid with type 1. We do have other people on the call who have type 1, but we are all interested in, when this cure is coming. They talk about it a lot, especially when you're diagnosed, when your child is diagnosed. It's kind of like the thing that doctors say to comfort you. And it's nice in the moment when that gets said, but 10 years later, you're like, well, the technology's gotten better, but the cure is still not a thing. So everyone is a little bit jaded, I think, maybe in terms of that. And this research that you've gone over is just fascinating. I think it's just amazing. But can you give us a sense of the time scale? I mean, I know that in 2000 was when this big breakthrough happened, but in terms of the work that you're doing, you talked about a lot of progress, but this was over the 5 years that you've been in the Ma Lab, and then the unfair part of the question is, what is the time? I'm not going to ask you when is diabetes going to be cured, but in terms of the rate of progress that you're making, what is your sense of solving these problems?
Well, it's an excellent question, and I think anyone who gives anyone a definitive answer is lying. this is the line I used to both you, Rob and Kelsey, when we discussed earlier. It's hard to say, you know, like I kind of joke around, if you have a mouse that has type 1 diabetes, you can give it to us and it's not a problem. But going in higher animal models, in larger animal models, it makes it much more challenging from a lot of different ways. And we're addressing these problems, and it's hard to say what the timeline is. I will say that I'm really encouraged by our recent progress, which I'm cryptically not allowed to talk about because they're not public record or patented. I will also say that the development in the New York Times, that was published in the New York Times, could be monumental, even if it means that there's so patients who receive so for a while, and this is now, I should clarify that this is my opinion and maybe not consensus, but for a while the limitation was really the cells. So making the best cells possible was a challenge, because that's a very challenging thing to do, is to take a cell that has no direction in life and then all of a sudden is an insulin secreting powerhouse. And now they're quite comparable to normal cells. And so that's a really encouraging development, is that, and again, their clinical trial isn't settled, but just at one patient, it gives a lot of hope. And not only does it give a lot of hope for the prospect of islet therapy, so injecting islets into the liver, stem cell based islets into the liver without encapsulation and with immunosuppression, but it means that many people like our group are going to have shots on goal, because now the bottleneck isn't having the therapeutic material, it's making a system that isn't recognized and destroyed by the immune system. And it's a surprisingly challenging problem, but one of the two big pillars has been conquered, may have been conquered, I should caution. And I'm sorry, I don't want to put, like, I don't want to say 1, 5, 100 years. Like, it's really hard to know, because anytime you go into a human patient, even going from really similar animals like monkeys, it's hard to say what will happen. And there's also the other component, which is regulatory and all of that, you know. But yeah, I hope that's an answer that's acceptable for everyone, even if it's, you know, not the you know, I can't sell snake oil. You know, I can't say that there'll definitely be a solution tomorrow. That would be unfair.
No. And that's what makes you credible, I think, for all of us. Actually, it's nice to hear you say that, and to put it in perspective that, you know, a very large challenge has not been solved, but is not the big challenge anymore. Maybe this, I have one, if no one else has another question, I have one question just to clarify in terms of like the immune response to these beta cells that you insert in a body. So people with type 1 it's the autoimmune response that destroys the beta cells in the first place, right? So that you know, before you get diabetes, you have these healthy beta cells, and then the body destroys them. Is that still a problem if you insert new beta cells? You know, if you have type 1, does your autoimmune system like still want to just destroy those? Whereas if it's somebody without type 1 and you inserted a packet into them, would those cells not be then at the same type of risk?
That's an excellent question too. So I'll say this. So yes. So and I'm going to wade a little out of my area of expertise. I actually didn't really discuss my true sub field of expertise, which is modeling, like doing mathematical modeling on all this stuff. But I'm going to just describe a little bit of the physiology, and maybe there's some doctors in the audience who could come in and correct me when I'm wrong. But what is recognized in type 1 diabetes are different molecules inside the beta cells that are really critical to the function of the beta cells themselves. So what I mean by that is during the disease progression, all of a sudden, for some reason, the body starts recognizing like insulin or insulin transporters inside the beta cells, which now make them targets for destruction by immune cells. And so if you put even beta cells from your own self that you just engineered from your skin, you took the skin cell and you turn it into a stem cell, you turn it into a beta cell, then you put it back into the body. That beta cell or stem cell beta cell will have all the same components that the body originally recognized as needing to be destroyed. And so you can't just do that, unfortunately. And this was why there was an excellent question, which is, can you just take your own stem cells and then put them in the body? The answer, unfortunately, is no, because they'll be destroyed by the same autoimmune mechanisms that destroyed the beta cells in the first place. And the second point is, you asked if you could put that in a non-diabetic individual and would they survive? And a theme that should have been driven home is that the body is just so creative, the most creative thing at recognizing stuff that's not supposed to be there. And so if you take cells from someone else and then you put them into you, even if you don't have type 1 diabetes, you'll have what's known as the allogeneic immune response. It'll just recognize that it's cells from another person. And so I suppose that's how I'd answer that question.
Well, it is after 8, and I promise not to take too much more of your time because Alec is actually in Ithaca, New York right now, so it is very late for him. So it's been that much more generous for him to spend his time with us and prepare this and answer our questions. It was a real privilege to get to talk to you and to have you on here, and we deeply appreciate it. And we hope that you do come back and we do stay in touch with you, and maybe we'll do this, every year or two, or whatever you're available to do. We would do this with you every month, but
I really love this. I really did, because it's a thing that we don't do in academia. First of all, we just don't really know how to do it. And maybe, professors may have more opportunities for this, but I would never have, Minglin, my advisor, was very generous to pass this on to me, and maybe to the detriment of you guys because he's more brilliant than I am. But I appreciate this and I really like it. And I think I learn, in a way, I learn more from doing this than from just talking with other people in my field. So I really appreciate it.
Well, we appreciate you. And if you would like to, one of the things that was surprising to me is that Alec asked us a lot of questions. In fact, he got with Kelsey separately and asked her a lot of questions about living with type 1 and some of the technology she uses and the trade-offs that she might, would you give up your pump if you had this inserted in your body and it had to be charged? And, there were some great conversations, and maybe we'll circle back around and do that one sometime where, we'll reverse it and we'll be your guest. And you can ask us anything you want, but we'll record it and put it on YouTube. So
nothing would make me happier.
Awesome. Well, we'll talk more about doing that then. Thanks to everybody who's come in here tonight, and please continue to support this effort and join us for different conversations about raising a child with type 1. We do it every third Thursday. So good night, everybody, and thanks for coming, and we'll see you next time.